So before we take a look at where the information from those MSNs flows out, let's just take a look at a picture like this, which shows information flowing in, right? So here is one of those medium spiny neurons, one of those MSNs that would be found in the putamen or the caudate. And you can see that most of the input comes from the cortex, right? From these cortical pyramidal neurons, right? And we said that this is excitatory input. We can add some detail in here, right? That here's this dopaminergic neuron, and we mentioned that this can be from the, maybe projecting from the substantia nigra pars compacta, right? And this is going to release dopamine. And notice where the synapse is made, right? The synapse is made right near where the cortical neuron is making a synapse, right? So the proximity matters here. The idea is that this dopaminergic neuron of the SNPC can modulate the effect that cortical output would have on this medium spiny neuron. Okay, so I think that's an important idea. All right, so let's look at those basal ganglia projections to other brain regions. So within the basal ganglia, right, the medium spiny neurons of the striatum, synapse, boy, I butchered that. Onto, and you can see here, the globus pallidus or the substantia nigra pars reticulata, right? So that's what this purple neuron is here. But importantly, this is a GABAergic synapse. That is, the medium spiny neurons release GABA, okay? So these are inhibitory. These two, the GP and the SNPR, the globus pallidus and the substantia nigra pars reticulata, we'll think of them as pretty identical. This one is, the SNPR is involved in vision. We're not going to get into that too much, but I think it's worth mentioning that these things, right, both of them, are similar cells. They perform similar functions, so comparable neurons. Similar functions. However, the part of the body that's controlled is different. The SNPR is going to modulate activity of the eyes. The GP is going to modulate activity more in the limbs and the trunk. So we're going to focus on the globus pallidus much more. Okay. Neurons leaving these areas this is what gives rise to the ability of the basal ganglia to influence activity. Okay, so it's going to leave the globus pallidus for our intents, and we'll see where it goes, right? Now, the globus pallidus gets split, right? And so that's what they show in this picture. There's an external portion and an internal portion. So here's the medium spiny neurons, right, which are in the striatum. And you can imagine that they may have some cortical projection out here, right? So there's the cortical neuron that's projecting on, and the dendrite of the cortical neuron that projects to the medium spiny neuron. This is glutamatergic, activating the neurons in the putamen and the caudate, GABAergic, and then leaving, right, when they don't show where these outputs go. Let me see if that's in the next slide. Yeah, they do show it. So let's see, outputs, goodness gracious, I'm getting overexcited here. Output of globus pallidus go to two different places, right? And so we're looking right in here. There's the globus pallidus. The first place they go is to the ventral anterior and ventral lateral nuclei, part of the thalamus. So we'll see this abbreviated VAVL. I know that this is thalamic. Okay, so you can follow one of these outputs here to the thalamus, and that the second output goes to the subthalamic nucleus. Yes. And what you have to note here, so here's the subthalamic nucleus, and we'll talk about what that means in a minute, but note that the output is coming from different regions of the GP, right? So if we go back here, what you can see, right, is there's both an external and an internal segment, and different output flows differentially to each of these regions. So this is via the internal segment. Oops. And so we'll abbreviate this GPES, and information flowing to the subthalamic nucleus, that's the external segment. And this is going to matter. This is going to matter pretty quickly. Now, again, I haven't mentioned where information is going from the substantia nigra pars reticulata. Those neurons project to the superior colliculus, right? That's this area here. But we're not going to talk about this pathway very much, right? So you see that we've got caudate, it's substantia nigra pars reticulata, to superior colliculus, which is going to allow you to move your eyes and your neck. But that's not the pathway we're going to focus on. However, what I will say is that both of these pathways, whether it's via the internal or external, did I write internal twice? My gosh. There we go. Sorry about that. So internal segment, external segment, external segment. It's a lot to keep straight. Okay. I think, where was I? Okay, that the globus pallidus output is also inhibitory. Okay, so we'll see that it's GABAergic as well. And so what does it mean? What's the output mean? It means that the effective excitatory output to the striatum is going to be inhibiting tonically active globus pallidus output. And this creates a disinhibition. So I think this is pretty complicated, and let's take a look at in more detail, excuse me. All right, so here's disinhibition in this circuit, right? Whatever neuron A is receiving excitatory input, okay? And because of that, it generates action potentials and releases an inhibitory neurotransmitter, okay? So we have this synapse here that's inhibitory. Now, this neuron B, is tonically active. So do you remember when we talked about neurons that don't need a stimulus to fire? Neuron B is tonically active, okay? And it produces inhibitory input when it's tonically active. So if we excite this cell, right? If we excite the cell and inhibit it, that disinhibits this synapse and allows it to fire. Okay? The result of that is this neuron can now release its excitatory neurotransmitter, and that activates lower motor neurons. Okay? So let's go through this in a little bit more detail. Let's look at the situation at rest first. And we'll use actual parts of the basal ganglia. Okay? So at rest, we can think of the striatum and the medium spiny neurons within the striatum as not generating any action potentials. Okay. Why? Because there's no input coming into it. Okay. And so because of that, and so if this is number 1, number 2 would be that neuron B, that is a globus pallidus neuron, this neuron is tonically active. The medium spiny neuron from the striatum doesn't do anything, right? So it produces GABAergic output. The GABAergic output inhibits, in this case, its cells of the thalamus. So this output the ventral anterior and ventral lateral thalamus. Okay, so if there's no activity coming out of the ventral thalamus, because it's inhibited, it produces no output, and we get no movement. Okay. Let's flip the script. What happens during a period of activity? Well, in this case, cortical projections excite the medium spiny neurons of the striatum. Okay, and these neurons release GABA onto neurons of the globus pallidus. Okay, so this synapse here was excitatory, mediated by glutamate. So the medium spiny neurons, though, release GABA, right, which is an inhibitory neurotransmitter. And so what this does is it inhibits the output. The tonic output of the globus pallidus. I didn't mention that, but in step 2, right, these medium spiny neurons releasing their GABA, they're releasing it onto the globus pallidus. So that tonic activity, right, because the globus pallidus neurons are spontaneously firing, and when they're inhibited, they'll no longer spontaneously fire. This inhibition of GP neurons disinhibits those VA, VL thalamic neurons, and so now it can excite an upper motor neuron. Okay, and so this synapse between the upper motor neuron and the thalamus, right, the VAVL of the thalamus, this is excitatory, right, so this is glutamate. And so you can see down here what we're saying, right? A is at rest. There's no activity in the striatum, so B is still firing action potentials. It's tonically active. Because this is GABAergic output, it inhibits the VAVL of the thalamus, and that inhibition produces no output, right? No motor response. But when the cortex excites striatal cells, right, those medium spiny neurons generate action potentials, and they release GABA. That GABA inhibits this tonic inhibition. Right? So we have inhibition of inhibition. That's disinhibition that we see here, right? So now we're sort of releasing the shackles from the VAVL neuron, and now it can generate action potentials, which are then passed on glutamatergically, so to speak, to the motor neuron shown here. Okay, disinhibition.